are some genes which we seem to be very tolerant of changes in them. We can kind of put up with their being a little bit out of whack, either because we've got maybe another gene a little bit like that can do some of the same job, or maybe we have a lot of it in the cell, so that a small change in the amount of that particular RNA doesn't make a lot of difference.
Fellow Homo sapiens, my name is Torie Robertson, and welcome back to Epilepsy Sparks Insights. Now, who knows about microRNAs and their relation to worms, how they were discovered, and how they are involved when it comes to the epilepsies? Well, today we do hear all about it from a person who can apply his knowledge of neuroscience, molecular neurophysiology, pharmacology, and genetics to figure out the answers for us, David Henshall. Please don't forget to share your thoughts on this episode with us in the comments below, because I really do enjoy reading your thoughts and responding to them too. Do subscribe so that we can educate the masses and empower way more people about the epilepsies around the world, and indeed more clinicians with patients who have an epilepsy, to provide the best care possible.
My name is David Henshall. I'm a professor at the RCSI University of Medicine and Health Sciences, which is a big medical school in the heart of Dublin in Ireland. And I do research on epilepsy. The area I'm most interested in is understanding what happens to genes in the brain during the development of epilepsy, and that's what we do research on, trying to find new ways to treat epilepsy.
When it comes to genes, obviously a rather complex topic in itself, I have to keep reminding myself, what is the difference between different types of RNA? Could you just give us a bit of a lowdown for people who aren't aware, haven't heard of them, or have forgotten, like me? What are RNAs and what are the different types?
What I could do is try and tell you the simple gene pathway, if you like, and kind of tell you where RNA comes in that pathway. So when we think about a gene, what we're usually talking about is a sequence of DNA that has the instructions to make a protein. So the way that this works is the DNA gets read in a process called transcription. And this produces an RNA, which we call an mRNA for messenger RNA. So this is a long RNA molecule, ribonucleic acid, long RNA molecule. And then that gets shunted out away from the DNA, out into another part of the cell, where it meets a big structure called a ribosome. And within the ribosome, the messenger RNA is read, and amino acids are glued together into a long chain, which forms a protein. So the gene pathway is sort of DNA, messenger RNA, protein.
You say protein, I just think of slabs of meat or something. So what's the difference between the protein we refer to in a slab of meat and the protein that you're talking about?
They could be one and the same. So one of the types of proteins that we have is the proteins that make up the muscles in our cells. Okay, but proteins can do all sorts of different things. So some proteins are enzymes that produce energy in your cells. Other proteins, and this is maybe most relevant to the brain, other proteins produce neurotransmitters, which are released by nerve cells when they communicate. We also have proteins that form little channels that allow the movement of molecules into and out of the cell. One of those molecules is sodium. Sodium, just like the salt that you put on your food,
or shouldn't put on your food,
and sodium moves in and out of cells during the firing of signals in the brain. In fact, it's one of the most important things, and actually one of the best known genetic forms of epilepsy is caused by a fault in the gene that makes that sodium channel.
Which form of epilepsy is that?
Dravet syndrome. Dravet syndrome is mainly caused by a fault in this gene called SCN1A. And so that gene, that encodes a protein that forms one of these sodium channels on nerve cells.
What are the benefits of these RNAs when it comes to us as the animals that we are? And, looking at evolution over time, did they always exist within organisms more or less all the time? Or are they... Because they seem to be a bit annoying, to put it politely, sometimes when it comes to epilepsy, because they, they play around a bit.
So yeah, there's lots of different types of RNA, right? The one that most of us talk about is mRNA. Most of us knew that name from the COVID vaccines, right? And the mRNA is the type of RNA which encodes a protein. But there's all of this other part of the genome that can be read, make RNA, but that RNA doesn't make protein. That RNA stays as RNA and does other jobs in the cell. So it's a bit like a gene. So there's a sequence of DNA that can be read. It makes an RNA, but the RNA doesn't then go on to code for a protein. It stays as an RNA, and then the RNA does jobs within the cell. And microRNA are one of the key types of those RNAs.
Okay, this needs to get more complex. So what exactly do these RNAs have to do with whether a person has an epilepsy or doesn't have an epilepsy, and how are you using that knowledge in your research? Tell us about that.
RNA is a measure of gene activity. So the RNAs that we were talking about, messenger RNAs, that code for proteins. So if you have epilepsy, there may be a fault in the sequence for that RNA, and so that RNA maybe doesn't make the protein that it should do. And so then you can end up with problems in brain function and for example seizures. But there are also other types of RNA that we're learning more about, the type that doesn't code for protein. We call that non-coding RNA. And there are lots of different types. Some of it's quite long, some of it's sort of shorter. And those non-coding RNAs are really important for regulating the gene pathway. So they sort of serve to either activate it a little bit more or to dampen it down a little bit less. And so if you have changes in that type of RNA, if you have changes in non-coding RNA, that can also cause problems. It can lead to too much gene activity or too little gene activity, the wrong things being switched on or off.
We're pretty sensitive creatures, right? With all these little things that could go wrong or malfunction.
Yeah, but it depends a lot on the gene. So there are some genes which we seem to be very tolerant of changes in them. We can kind of put up with their being a little bit out of whack, either because we've got maybe another gene a little bit like that can do some of the same job, or maybe we have a lot of it in the cell, so that a small change in the amount of that particular RNA doesn't make a lot of difference. But then there are other types of RNA where small differences make a huge impact on brain function. And actually, the SCN1A gene is a nice example of that, because that gene is particularly important for the firing of our inhibitory neurons within the brain. And anything changes in how they fire, then we start to have problems.
I went to a conference and I met lots of people with SCN2A and SCN8A and mutation, and one of them was a data scientist, and everybody else had severe intellectual disability, autistic traits, mostly non-verbal. And I just thought, wow, this really shows you can have what appears to be the same on paper, at least to the layman, but the variety of how that can impact people is huge.
Yeah, absolutely. And we're only just starting to sort of understand or make sense of, why a mutation in one particular gene can cause something really catastrophic, whereas another mutation or variation can be really well tolerated. A lot of it comes down to, so for some of the proteins that are important for, movement of molecules inside and outside of brain cells, there'll be a particular amino acid that sits right at the center of it, that sort of acts as a gateway. And if you get a mutation there, then the entire protein stops working properly. So there are sort of some areas of genes that can sort of handle a bit of variation, in other areas where any kind of variational mutation can completely damage the function of the gene.
And so the type of research that you do you look at a particular gene that is that commonly causes epilepsy? And we'll get onto your marvellous book in a moment. But or are you looking at the whole spectrum of things? Or
so we don't tend to concentrate on a single gene or a single cause of epilepsy. In fact, the area of epilepsy we work most on is temporal lobe epilepsy, which is it's a very common form of epilepsy, particularly in adults. It's very often drug resistant or difficult to control seizures with. And we don't see a very strong genetic component to that type of epilepsy. It's generally understood that epilepsy has resulted from a combination of environmental factors, maybe things that you experience growing up, mixed together with your genetics and perhaps something else that we still don't fully understand. So the sort of working hypothesis for our lab, as well as several others on temporal lobe epilepsy, is that actually what's happening there is there are multiple different changes within the brain, maybe changes to neurons, changes to their support cells called glia. There's maybe an inflammatory component, maybe something to do with the blood supply, the sort of energy balance within that tissue. And so you've got a lot of things changing a little bit, and that makes it particularly challenging to fix. As an epilepsy, we don't think you can just go in with a sort of a one gene approach. And that was one of the reasons that I became really interested in the area of microRNAs, for reasons that I can explain.
do these things just faff about in your brain, or are they like going to other organs and other tissues?
They're in every cell in the body. So every cell produces microRNA. The brain produces the most microRNA molecules and the most different types of microRNA. And one of the reasons is that the brain is, in terms of its complexity, in terms of the different cell types, it's absolutely packed with these microRNAs, which are very important, in fact, for producing all of the variety, the rich variety of brain cell types. But they can be detected in other places, so they can be detected in the fluid that surrounds and circulates through the brain, called CSF, or cerebrospinal fluid. You can detect them in the blood. You can also detect them in things like tears and saliva. So there's a whole area of research on microRNAs as potential diagnostics. And one of the reasons that we think they might work is that we know that particular cells generate very particular microRNAs. So if you imagine there's a microRNA that's only produced in one part of the brain, if you can detect that in the blood, that could tell you straight away that there's a problem with that part of the brain if no other part of the body makes it.
Right.
And microRNAs are also, they're quite sort of chemically stable, so they can survive quite a long time in fluids. And that's an important factor to consider for sort of practical use of these things. You know, if you want to take a test in a hospital, you know, that molecule has to survive for a while in that fluid. So there's a couple of really exciting stories that I cover within the book about particular experiments that were done. So I could mention a couple of them now. One of my favorite experiments was an experiment that demonstrated the effect that even a single microRNA can have on a cell. So a group of researchers took one of the microRNAs that's only made in the brain, and they put it in a common cell that's used in most labs around the world. It's called a HeLa cell. There's a story around the origins of HeLa cells that I could tell you a little bit more about, but essentially this is not a brain cell, okay? So they took one of these microRNAs that's made only in the brain, and they put it into this other cell type, HeLa cell, and then they looked at what happened to the gene activity, and it reduced the level of all of these genes that are not made in the brain. So what this told us was that what this microRNA is doing is keeping brain cells, and stopping them make things that shouldn't be in brain cells.
Which is a good thing, right?
Which is a very good thing, yeah. And there's a large amount of this particular microRNA which is continuously made inside neurons within the brain. and it stops other non-neuron genes getting made in that cell type. And if you remove that microRNA, then the genes begin to sort of bubble back towards something that isn't neuron. So that was one of these really cool experiments that was done really early on.
So like without this microRNA, we could potentially have like any old weirdo cell growing in our brain.
Yeah, without that particular microRNA, and there's one or two others that are a bit similar, our neurons wouldn't have all of the properties that we need them to have. So it's sort of like leaky genes would start to come through, and then the microRNA is just like, no, not on my watch.
So the microRNA is the boss of what goes down. And what was the next one you had to share?
So one of my other favorite experiments was to try and understand what controls the amount of microRNA in a particular brain cell. And what they did was they connected a little colored substance onto a microRNA, and they watched with a microscope little flashes of that color as that microRNA got made. And what they learned was that microRNA was getting made just as a signal came through from one neuron to the next. So the production of a microRNA is actually connected to the signals between brain cells. And this is really important for things like learning and plasticity and memory, because we know that brain cells can change shape slightly in terms of their connections, and that's an important thing, that those connections can strengthen as we learn things. So what their experiment showed was that actually microRNAs are really important for that strengthening process. They need to be able to switch on and off, and by doing so they can strengthen or weaken the connections between brain cells.
So something goes, an experience happens, something affects one particular part of your brain cell, and the microRNA that already exists will go, yo, I'm here or not, depending upon what is happening in your brain at that time.
Exactly. So to sort of learn and to make memories, we know that new proteins need to get made. And so microRNAs are sitting right at the center of that process, controlling it all and making sure that it happens smoothly and correctly.
That is so cool. It's kind of scary cool, but very cool. Yeah. Okay, and we'll hear about that in the book, right?
Yes, yeah. There's a good description of that experiment, as well as the other experiment with the HeLa cell.
Thank you so much to David for managing to explain to us what microRNAs are, their significance, and giving us an enticing sliver of his new book. Do make sure that you join us next week for part 2 of 2 with David, where he should be telling us much more about his incredible discoveries, microRNA therapy today, and exciting us more about his epilepsy research going forward. Do check out David's book on the website torierobinson.com, where you can also access the podcast, the video, and the transcription of this episode. And if you haven't already, don't forget to like, comment, and subscribe to the channel. Share this episode with your friends, colleagues, family members, and see you next week.